Today we will be learning about sickle cell anemia. This is one of the Mendelian disorder that we have learnt in our 12th class. So let us discuss it in detail today. Sickle cell anemia, it is a disease where the red blood cells it convert into sickle shaped, right? Sickle shaped. The normal shape of the red blood cells is biconcave, but the diseased person, they have the red blood cells in the form of sickle shape, and these type of cells they cannot stay alive for more than 10 to 20 days. So they die quicker than the normal RBCs. That is why the bone marrow has to continuously produce this cell, and as such the patient have a constant anemia problem in the body. That means since the RBC get die very frequently, so the number of RBC in the blood always remains less than what needs to be required in a normal person. So this disease is controlled by a single pair of allele, that is HBA genes. So in a normal person, so HBA genes it code for CTC, that means these are the three nitrogen bases, that is C, cytosine, thiamine, cytosine. So when this triplet it will be coding for the mRNA, then during the transcription process the mRNA has G, that is C will be producing the corresponding G, thiamine, adenine, and again C to guanosine. Now what will happen when this triplet codon will code for the amino acid, then this triplet codon it code for the glutamic acid. Glutamic acid is one of the amino acid, it code for this. Now the glutamic acid, we have to remember, in the polypeptide chain, which is one of the polypeptide chain which forms the hemoglobin molecule, it lies in the 6th position. So it is 1, 2, 3, 4, 5, 6. In the 6th position lies the glutamic acid in normal hemoglobin polypeptide chain. This so here what we see, this is a correct one that needs to be present in our hemoglobin, the polypeptide chain, right? So the normal genotype for this will be HBA and HBA. So this is the pair which is present in a normal person. So here I repeat myself, the gene which is coding for the normal polypeptide chain is the GAG, this, and here A, that means the adenosine jo hai, it's coding for the glutamic acid. But what about in a patient having the sickle cell anemia? In the patient who is having the sickle cell anemia, the patient have a diseased genotype, that is this HBS. So here in when the diseased person is having S here, so here in normal case what we find, it is CAC, right? So in this one it changes from thiamine to adenine here, right? In normal case we have found this is occupied by the thiamine position, this is a thiamine position, but in case of a sickle cell patient, what happened here, the adenine will take the place of the thiamine. As a result, the mRNA that is produced after transcription of this will have U here, the uracil here, right? Uracil here, because adenine is the correspondence to the adenine, we are having uracil here. Now this uracil is present in case of the sickle cell patient mRNA, that is the mutated nitrogen base here. So when this happens, so because of these uracil nitrogen base, this triplet, that is GUG, it code for the valine amino acid, right? It code for the valine amino acid. As such, the valine amino acids, it comes in the 6th position of the polypeptide chain in place of the glutamic acid. In this condition, what happened? The patient is now having a separate polypeptide chain that is present in the hemoglobin. So as such, for this, the patient now, he will be having or she will be having the RBCs as sickle shape, like this. It's crescent shape, or we can say it's sickle shape. Okay, so now the disease phenotype will be, now it's a homozygous, you see, homozygous condition, and here both the alleles is represented by HBS and HBS. Now how it is, the person is affected by this. Before showing the cross, let us first understand this is a autosomal recessive disease. This is a autosomal recessive disease because these alleles, they are not a part of the sex chromosome, so they are an autosomal chromosome. And now let us see the cross, how it gets transmitted from the parent to the offspring. So we are now what we are doing, let us consider this is the father and this is the mother. It's very clearly it is understood here both the parents, they are having heterozygous condition. See here, HBA HBS, here also HBA HBS. That means here both the parents, they are the carrier of the disease. So it is heterozygous condition. Now the gametes that are formed are HBA HBS, and similarly these two, the similar gametes are formed. Now when we make a cross in this way, what we find is that this, the homozygous condition here, this is a homozygous condition showing this is an unaffected, normal in person, you can say this is a normal person. He is not affected, neither he is a carrier of the disease. But in these two cases, you see where we are having HBS and HBA, here also in this case also, these two individuals, they will be carrying the disease allele in their genotype. They will not show the disease obviously, but they are the carriers. So these genotype, they are the carriers in these two cases. But what about here? See this condition, it's a homozygous. Individual, and here the genotype, that the both the alleles of the that is forming the genotype are affected. So this individual will be an affected person. We can say he will be a patient of the sickle cell. So what we have learned from this cross is that the sickle cell anemia will be transmitted to only the individual whose parents both will be a carrier of the disease. Both of them will be heterozygous individuals, and they will be apparently carrying the disease, but they will not showing the conditions. But after the cross, what we see here after the cross, we can very easily make out here only there is 25% of chance to be the affected progeny. That means the progeny, out of 4 progeny, if we consider, only 25% will have the probability to have the disease condition. Okay, that is all for today. Thank you.